Hello everybody, I am the pre-Dr. Bennett, and today I'm going to be changing up the format a little bit. Still on Khan Academy, MCAT prep, but now I'm in biomolecules, the section underneath where he does the passages, and I'm going to be focusing on just real content. I've recently been getting more into content review. I've been doing a lot of passage work, a lot of test-taking skills, and I want to focus a little bit more on content, and I figured I would try to go through all of the practice problems that are offered. So they're typically in sets of 10. They might all be in sets of 10, but at least everyone I've done has been a set of 10. And I'm starting from the top. I'm going with biomolecules, and I'm at amino acids and proteins. Okay, so unlike how I do the passage problems, in this in this type of video, I'm going to be going through all this stuff live. So there is no editing. You're getting my real thought process, real time, and all my rights and wrongs, so everyone can laugh at me and have justification for why I do as poorly as I am surely going to do on the MCAT. All right, so first question. Which of the following amino acids has a net negative charge at physiologic pH, around 7.4? Okay, so we have lysine, glutamic acid, asparagine, and histidine. All right, so this one really helps if you have taken if you've taken biochem. If you haven't taken biochem, I would suggest you just don't take the MCAT. So at physiologic pH 7.4, so what we're trying to see is, okay, if we're at pH 7.4, is that going to have deprotonated everything to the point of giving it a net negative charge? So, okay, lysine at pH 7.4, you're going to have a negative charge on the carboxyl terminal. You're going to have a positive charge on the amino terminal, I think. Is that a zwitterion? But you're definitely going to have a positive charge on that R group. So I think that one's ruled out. Glutamic acid, so that's a negative on the R group, and that's going to have an overall negative charge, I think so, right? Asparagine, that's going to be like lysine, that's going to be a positive charge on that R group. Histidine, also a positive charge on the R group. I'm going to go with glutamic acid, and yay, I love the little Khan Academy noise. Makes me feel so good. I wonder if they did psych-soc research into that. It's applicable, people. All right, next question. A polypeptide with a net positive charge at physiologic pH around 7.4 most likely contains amino acids with R groups of what type? So if you have a net positive charge, we're at physiologic pH, what type of R groups do we have? Is it aromatic R groups? I can almost surely write that off as no. Is it basic R groups? That could very well be it, because basic R groups are going to want to grab up excess hydrogen, and they normally only will release that hydrogen around their pKa, which is typically going to be up like past 9, past 10 in some cases. Allopathic R groups, allopathic. I know amphipathic. Allopathic, I think I've known that word before. Maybe that just means amphipathic, but for proteins. I might be saying a bunch of gibberish, but I don't think that's the right answer. And then acidic R groups, that's the opposite. I would expect basic R groups. Cool, we got it right. Okay, third question. The amino acids in hemoglobin or any protein uniformly have which of the following configurations? So the amino acids in any protein. Oh gosh, I'm bad at this. Oh, I forgot this. I haven't taken biochem in a while. It's either D or L. Oh, help me out, people. I'm going to say L. I was right. Okay, but let's see why I was right, because that was a guess. Okay, so the first hint. Configurations, either relative or absolute, in amino acids refers to the stereochemical configuration around the chiral carbon, right? Due to the differences in priority of different amino acid side chains, not all amino acids have the same absolute configuration, which refers to RS naming convention. Some amino acids are R and some are S. Okay, that makes total sense. However, all amino acids have the same relative configuration, which refers to the DL naming convention, which is, I remember you look at that when you see Fisher projections. I think. Or maybe it's after you've completed the ring in sugars. I kind of remember that. But this might be a whole different configuration rule. But it's something I'm going to have to go back and look at because that's important. So the relative configuration is D and L. All biologically produced amino acids are in L configuration. Okay, question 4. Which of the following properties of a protein is least likely to be affected by changes in pH? Okay, primary, tertiary, secondary, or net charge. So net charge, right out. Of course, that's going to be affected by changes in pH. Primary structure, I doubt it. Highly doubt it, because those are peptide bonds. Maybe with really crazy changes in pH. Secondary structure. Yeah, right. Hydrogen bonding and alpha helices. I know that much at least. And in beta sheets, I think it's all coordinated by hydrogen bonding. And that could totally get messed up by pH changes. Okay, and then tertiary structure, 100%. So I'm going to go with primary structure. And yep, we were right. So primary structure does not get changed by a lot. It takes a lot to break those peptide bonds. Okay, hydrogen bonding between separate subunits of DNA polymerase is an example of which of the following? First degree structure, second degree, third degree, or fourth degree structure. So keyword here is separate subunits of DNA polymerase. So they're talking about quaternary structure, 4-degree structure. Cool, got it right. All right, the unique cyclic structure of which of the following amino acids plays a central role in the formation of alpha helices and beta sheets? Okay, the unique cyclic nature, and the answer choices are lysine, arginine, valine, proline. Well, they kind of gave it away by saying the unique cyclic structure. I assume the answer is proline. That's what I clicked. Yep, it's right. Okay, so the other ones, but proline, so plays a role in formation of alpha helices and beta sheets. I could see if you have turns, proline is really useful because of its bizarre structure to create turns. Kind of like glycine as well. Okay, question 7. Electrophoretic separation of leucine from a protein sample would be least effective at which of the following pH values? Okay, electrophoretic separation. I don't know what oh no, electrophoresis, gel electrophoresis. So you are looking at charge, right? So R group stuff. Okay, of a protein sample be least effective at which of the following pH values? Electrophoretic separation. I'm looking at all these pH values. It's 2.4 acidic, 1.4 acidic, 0.4 holy crap acidic, and 7.4. That is physiologic pH. I'm just gonna click it, and it was right. Okay, let's see why. Okay, hint 1. Leucine has an allopathic side chain. Correct. Oh, that's what allopathic means. Aha, it made sense once I saw it. Okay. At physiological pH, leucine exists as a zwitterion. Okay. Yeah, so it would not be affected by, if you're at physiological pH, you have a zwitterion, it's not going to get pulled by gel electrophoresis. Electrophoretic separation of leucine from a protein sample would be least effective at pH of 7.4. Cool. All right, question number 8. Electrophoretic separation at pH 6 of a sample of polypeptide 1, which has a molecular weight of 100, polypeptide 2, which has a molecular weight of 200, and polypeptide 3, which has a molecular weight of 400, would result in which of the following? Note the isoelectric point of each peptide occurs at pH 6. Okay, so if the isoelectric point happens at pH 6, you're not going to be able to separate them based on pH. You have to separate them based on size. So if they're all at their isoelectric point and you're separating based on electrophoresis, I think based on the last question, they wouldn't move, right? Cool, that is the correct answer. If we're talking about electrophoresis, people, it doesn't matter how big they are. Well, it might matter once you start pulling them, but if they don't get pulled in the first place because there is zwitterion, then you don't have to worry about that. Okay, question 9. The alpha helix is an example of which of the following structural properties of proteins? Okay, alpha helix, typically seen in transmembrane proteins. I do a lot of work with transmembrane proteins, so I'm very familiar with this. Alpha helix is a secondary structure, that is typically found in membranes as a transmembrane region. For something like a GPCR, has the classical 7 transmembrane region. You have voltage-gated ion channels, which have that repeat region of 6 transmembrane segments. All of those transmembrane regions are alpha helices. They are secondary structures, a coiling of the primary structure to create a more hydropathic environment. I don't really know a better way to describe it, but I'm just going to go ahead and click secondary structure. And that was correct. Okay, number 10. All hydrophobic amino acids, valine, leucine, isoleucine, etc., share which of the following properties? So hydrophobic amino acids, what do they all share? Basic R groups? No. Acidic R groups? No. Polar uncharged R groups? No. Non-polar uncharged R groups? Yes. Easy peasy. Ah, we got 10 out of 10. Strong work, gang.